Thank you everybody for joining today's session. This session is titled Computational Approaches to Understanding Psychosis. Each speaker will present their slides and I will moderate a brief Q&A portion at the end of this session. If you have additional questions after watching each of the speakers, we recommend emailing them with your questions. Be sure to promote your science, colleagues, institutions, and SIRS by posting comments and pictures to Facebook and Twitter. Use the hashtags SIRS2020, SIRS, and schizophrenia. And don't forget to retweet, repost, like, and share, and comment on other social media posts. Today, we have three outstanding speakers: Dr. Tiago Maia from the University of Lisbon, Dr. Andreas Meyer-Lindenberg from the Central Institute of Mannheim, and Dr. Kirsten Ritter from the Charité University. I will now turn it over to Dr. Maia for our first presentation.
Well, thank you very much, Deanna, for the kind invitation to be here and have an opportunity to share with the audience my perspective on the role of dopamine in schizophrenia, which I'm very, very happy to do. Now, I should start by saying that I don't really do empirical work on schizophrenia, and really sort of the extent of my contribution to schizophrenia has been this article that has been this article that I wrote with Michael Frank a couple of years ago, in which we tried to provide an integrative perspective on the role of dopamine in schizophrenia. That's where I'm actually getting the title of my talk from. And basically, we tried to bring together a lot of ideas to try to provide a fairly comprehensive explanation of schizophrenia. So, of course, this is a little bit old news by now. So what I'll try to do is I'll try to briefly take you through this, but then I will also look at more recent evidence and see how did the ideas that we suggested hold up in the interim, and can we use these ideas to make more sense of the treatment, particularly of negative symptoms, than we had room to address in this paper, and hopefully that will be of interest to many of you. And as you'll see, there's a lot that we still don't know and a lot to be done. And my hope is also that this will encourage people to work on a lot of the questions that I think are outstanding and that are really important. So since this will be very much a review type of talk, I thought that I'd start with one of my favorite quotes about science from Henri Poincaré, who said, "Science is built up of facts as a house is built up of stones. But an accumulation of facts is no more a science than a heap of stones is a house." And so basically, of course, science is about facts, but it's also about bringing those facts together into a coherent picture. That's what I'm aiming to do here with you today. So hopefully that will be of interest, and you'll indulge with me as I do sort of a review type of talk. So let's start by establishing a very basic fact, and this has now been shown in several meta-analyses. This is the latest one that I'm aware of, showing that the schizophrenia clearly involves striatal hyperdopaminergia. So there's in the striatum, there's increased dopamine release capacity, increased dopamine in the synapses, and increased dopamine synthesis capacity in schizophrenia. And so this is very well established. It's as uncontroversial as you'll get in psychiatry research. Now, what's interesting, and as I was working on that paper that I mentioned with Michael Frank, I had this clear paradox in my mind that was really, really bothersome to me until I was able to figure it out. And it's this big paradox, which is that everything we know about increased striatal dopamine from the animal literature, from work in healthy humans, and even from other disorders, is that increases in striatal dopamine lead to improved learning and processing of rewards. So better learning from rewards, better signaling of positive prediction errors in the striatum, more striatal activation during reward anticipation, willingness to exert more effort to get rewards, and more generally, psychomotor activation. So what's the conundrum? The conundrum is that, of course, as you'll know, in schizophrenia, it's the very opposite in all of these domains. So these findings are now very well established. And essentially in schizophrenia, there's a reduction in these processes rather than an increase. And the conundrum, of course, is that if schizophrenia involves increased striatal dopamine, how is that possible? What is actually going on? These deficits in schizophrenia have been found to correlate fairly consistently with negative symptoms, as you would expect. So if you can't learn from rewards, if you can't anticipate rewards, if you're not willing to exert effort for rewards, that's the kind of thing that has a lot of face validity to give you negative symptoms. So this part is clear. What's not clear is how it is that in schizophrenia, you have increased striatal dopamine and yet you have these deficits. And really, I had sort of the insight that I had into this came when I saw what amphetamine does, particularly at high doses to the brain, and particularly to dopamine release. And so it's very interesting that amphetamine at high doses increases spontaneous transients. Transients are these sort of overflow of dopamine in target regions, in this case in the striatum. And at a high dose of amphetamine, you see that dopamine is sort of going up and down, up and down. It's being released spontaneously in a way that's not tied to events, it's not tied to rewards or to anything that's relevant. And so this, of course, is the kind of thing that could give rise to positive symptoms, because you're assigning value to things that you shouldn't be assigning value to because you're having these aberrant transients. Of course, this idea is not new. It has been around for quite a while. The idea that the chaotic firing of dopamine neurons and things of that sort could be what's giving rise to positive symptoms had been in the literature already. What I think is really interesting, and where amphetamine really helped at least me to potentially make sense of the conundrum with the negative symptoms, is that at high doses, amphetamine has an effect that is very surprising, which is that it actually blunts what I call adaptive transients. Those are the releases of dopamine that you get when you should get it. So when you get a reward, when you get a cue that predicts a reward, or more generally, when you get a positive prediction error. But at high doses, amphetamine actually blunts those signals. And that's shown here in this nice study in which here I'm showing the time where a cue that predicts reward is presented. And then there's this phasic release of dopamine, as you'd expect, under saline. If you have a moderate to high dose of amphetamine, you get potentially even an increase in that signaling. So especially it takes longer to come back to baseline, as you'd expect, because amphetamine is blocking the dopamine transporter. But what's really interesting is what happens if you give a really high dose of amphetamine. And that is that now, when you present the cue that predicts reward, there's absolutely nothing. There's no signal, no dopamine signaling, despite the fact that at this dose, there are huge amounts of those spontaneous transients. And so then really this was sort of my aha moment for this was, well, maybe then you have a lot of dopamine in your system, but it's not really being elicited by the right things. It's not being elicited by rewards, by cues that predict reward and so on. And that's what gives rise to negative symptoms. And that's kind of the general theoretical framework, the theoretical proposal in this paper that I mentioned in the beginning, and that I did want to bring you up to date on in case you hadn't seen it. And then in the paper, we go through how the reduction in adaptive transients for relevant stimuli, which essentially corresponds to reduced signaling of positive prediction errors. If you work through the effects of that through the equations of reinforcement learning, which I won't do here because that has been published, you can always look it up if you're interested. What that does is it ends up producing a set of effects, which are exactly those effects that seem to be paradoxical in schizophrenia, such as reduced signaling of positive prediction errors in the striatum and other regions, reduced striatal activation during reward anticipation, reduced learning from rewards, reduced effort for to get rewards, and all of those things that I had mentioned seem paradoxical. And then they all of a sudden make sense based on this very simple idea. Now, like I said, this has been published, and I wanted to now look into more recent things and see what else have we learned and how can we enrich these ideas. And I'd like to start by, I'll start by focusing a little bit on a recent study on the role of amphetamine in dopamine release. And this is a study in rats, but I think it's very interesting because it helps me to sort of reinforces that idea that I suggested initially, that amphetamine might be blunting the prediction error signaling, which I was based on that single study. And this study really provides evidence that's very, very consistent with that. And then it also provides a set of results that I think are very reminiscent of what goes on in schizophrenia. And so it helps also, it helps me to again establish the parallel between these two states, this high amphetamine state and what might be going on in schizophrenia. So in this study, which I'll describe a little bit, it's a very simple Pavlovian conditioning experiment in which there's a CS that's associated with a high probability of reward. It's the CS high. There's another CS, the CS low, which is associated with a low probability of reward. And basically, this is all that rats have to learn. And here are the findings, the behavioral findings. I'll focus here on the green part, just ignore the black one in the sake of expediency here. And notice what happens without the drug. As you would expect, the animals respond much more. So here in the pre-drug, the animals respond much more to the CS high than they do to the CS low, to which they respond just a little bit above baseline. Now notice what happens with the drug when they're giving amphetamines. In that case, the response to the CS high actually goes down dramatically. And this is exactly what you would expect if amphetamine is blunting positive prediction errors, because if it's blunting those adaptive transients which signal positive prediction errors, you're not able to learn the value of the CS high. You're not able to learn that it leads to reward, and so you'll start responding much less to it. And so this is very consistent with that idea that I suggested, that the high doses of amphetamine were blunting the adaptive transients. Now, what's really interesting to me in looking at these behavioral findings in some detail is that the rats not only respond a lot less to the CS high, but they respond a lot more to the CS low, which is kind of bizarre. But from a schizophrenia perspective, this is extremely interesting because there's a whole line of work on the so-called aberrant salience experiments. And what those experiments always consistently show is that patients with schizophrenia respond less to a relevant cue, like a CS high here, and they respond more to an irrelevant cue, like the CS low here. So this is very, very much like you see in schizophrenia, including in all of those aberrant salience experiments. Then, of course, this study, this is just the behavioral part. Then there's a whole part on activation with the measured using a special technique. I won't get into that, but essentially the findings again are very reminiscent of what you see in schizophrenia and very consistent with this idea that amphetamine is blunting adaptive transients. So here are the findings when you present the cue. As you'd expect, without the drug in the nucleus accumbens, you get this nice signaling when you present the cue that predicts reward, but that signal is very much blunted under amphetamine. And this is exactly what happens in schizophrenia, in which cues that predict reward elicit less activation than for healthy controls, or when you're anticipating reward, you have less activation. And then they also look at outcome. So what goes on here at the outcome. And it's a little bit more complicated because there's 4 cases. I'll focus just here on the case of the reward after the CS low. That's when this reward is really unexpected. And so it gives the largest positive prediction error. And so that's this case here, this nice, that's why this curve is much higher than the other ones. You have a bigger positive prediction error here. But notice that under the drug, that is completely blunted. And so this again is like what you see in schizophrenia at activation, at outcome. And also this is exactly what you would expect if there's blunted signaling of prediction errors, which by the way, they do modeling and they show that's the role of amphetamine is blunting the signaling of the prediction errors. Okay, so this is just I think it does help to reinforce all of those ideas that we had suggested and this parallel to the amphetamine case in a way that I think is quite interesting. Okay, but enough about amphetamine. Let's now move on to the treatment of schizophrenia. And particularly, I'll be focusing a fair amount on negative symptoms. And so if the account that I proposed is correct, then the goal for the treatment of negative symptoms should be to increase those adaptive transients, but we have to be careful not to increase the spontaneous transients because that might actually worsen positive symptoms. And so it's a little bit of a tricky balance here to be able to do one without the other one. And the truth is that we know very little about which drugs would be able to pull this trick. So to a large extent, I think that we've been flying blind in trying to treat negative symptoms. Here are two very interesting reviews, one including a meta-analysis, one of psychostimulant treatment of negative symptoms, another one on pro-dopaminergic drugs for treating the negative symptoms of schizophrenia. And so you might think that to some extent, this is consistent with my suggestion that we need to amplify those adaptive transients. And to some extent that is true, but it's also the case that we really don't know if these drugs do that, especially in schizophrenia. So we don't really know if psychostimulants and other pro-dopaminergic drugs are what I would call pro-adaptive transients, if they really increase adaptive transients, especially in a disorder like schizophrenia where there's already hyperdopaminergia. Truth is, we just don't know. It could depend on the dose, with high doses potentially being less effective. That's kind of the lesson from the amphetamine, the initial amphetamine study that I showed, where a low dose seemed to amplify the adaptive transient, but a higher dose actually blunted it. It could potentially depend on the patient. So some patients, or even most patients for that matter, might be on a state that's kind of like having too much amphetamine in a sense, that in their, meaning their dopaminergic system might be in a state like that, in which case psychostimulants, for example, even at a low dose, might actually blunt the adaptive transients and worsen negative symptoms, let alone positive symptoms. And so the truth is that we just know very little about these things. There's a lot that we don't know, and I think we need to do this work in the basic science to understand these issues much better. So having said that, I do want to now take some time to talk about two drugs that have been shown empirically to potentially be interesting for negative symptoms, and try to put them in this framework and see if this framework helps to make sense of them and if they're consistent with the framework. And so one of the drugs that seems to be promising from the meta-analysis that I mentioned in the previous slide is modafinil. And so I thought I'd ask the question, well, what do we know about what modafinil does to these dopamine transients? It turns out we know very little, but there's one study that is suggestive, although it has important limitations. And here's what the study finds. The study actually finds that modafinil indeed does increase these adaptive-like, phasic-like dopamine transients, both in the dorsal striatum and in the ventral striatum. And it increases them quite markedly. So to some extent, this is suggestive that indeed modafinil might be increasing the adaptive transients, which is what you might want to be doing. The reason the study is not super conclusive about this, well, there's two reasons. One is that this study is actually in anesthetized animals, and so these transients are elicited by electrical stimulation of the medial forebrain bundle. We don't know if the same thing would occur for cues that predict reward and rewards and so on in awake behaving animals. That's one thing. And the other thing that's very important is these are sort of normal rats. And so we don't know if you're already in a hyperdopaminergic state, if this would still occur, and that what doses it would occur, whether it would invert into an inverted U-shaped curve and things of that sort. In this study, they also looked at spontaneous transients, and of potentially some concern to positive symptoms, they actually found that modafinil, when co-administered with raclopride increased spontaneous transients, and this could potentially be of some concern. The reason I'm not too concerned about it is first, the authors themselves note that there's some limitations in generalizing this to the awake behaving case, even because of the effect of anesthetics themselves on dopamine neurons. And second, we need to remember that what matters is not so much the transients themselves, but how they affect postsynaptic receptors. And so patients with antipsychotics might be sort of protected from these transients because they have a high dose of the antipsychotic blocking the postsynaptic receptors. Whereas in this case, although they did co-administration with raclopride, the goal of raclopride here was actually to inhibit the presynaptic D2 receptors, and therefore leading to potentially more firing in dopamine neurons and contributing to the spontaneous transients themselves. So given that also modafinil has not been shown to really increase positive symptoms in that meta-analysis, I wouldn't worry too much. But again, we do need to look at these things in more detail, and there's a lot that we don't know about this. And I sort of already mentioned these things, so I'll push ahead. So I move on now to antipsychotics and their relation to negative symptoms. And this is from a recent review and meta-analysis of antipsychotics for patients with schizophrenia and predominant or prominent negative symptoms. And I highlighted here a sentence from the abstract, which I think is very, very important. We mentioned this in our paper too, which is this issue of the confound that occurs by improvements of secondary negative symptoms, which are very difficult to dissociate from the primary negative symptoms. But the main conclusion of this meta-analysis is that amisulpride is the only antipsychotic that outperformed placebo in the treatment of predominant negative symptoms. And so does this make sense in light of the proposals that I was suggesting? As a matter of fact, it does make a lot of sense, because amisulpride, there's lots of converging evidence that suggests that it might indeed be increasing those adaptive transients. So why? Well, at low doses, like in the studies that are included in this meta-analysis, amisulpride actually blocks mostly and strongly presynaptic D2 receptors, therefore having sort of a pro-dopaminergic effect. In fact, because of that, it's even used to treat dysthymia. And there's direct evidence for increased striatal activation for positive prediction errors, which is then predictive of reward learning in healthy controls under these low doses of amisulpride. And even in patients with major depression, there's also evidence that they have increased striatal response to reward under low doses of amisulpride. So this is all quite consistent with the idea that amisulpride is going to increase the adaptive transients. The direct experiment. Okay, so I just my internet just got cut off, so there might be a little hiccup here in the presentation. I apologize for that. But I was about to say that I had just the only study that I found that's really relevant to this is a study of sulpiride that shows that sulpiride increases evoked dopamine overflow actually quite significantly relative to controls here in the accumbens. Again, this study is somewhat limited because this was also through electrical stimulation and so on. But nonetheless, all of this sort of seems to suggest that really the effect of low dose of amisulpride should be to increase the adaptive transients as you'd expect. Okay, so we need to be very careful though about the dose issues, because at higher doses, amisulpride and sulpiride actually have the opposite effects. They impair reinforcement learning, striatal signaling of prediction errors, and even striatal responses to primary rewards, such as chocolate. And this occurs because at higher doses, they have strong postsynaptic effects, which then are sort of counterbalancing the presynaptic effects. A note of caution is that these studies involve acute administration, and there's some evidence that chronic antipsychotic administration has somewhat different effects. So we need to be a little careful about that. But nonetheless, this seems quite consistent. So I'll just summarize then everything I told you. The theoretical proposal is that schizophrenia involves a combination of increases in spontaneous transients with a decrease in adaptive transients. I have a single arrow here, meaning that this is not hypothesized to be two deficits, but two phases of the same deficit, as you have with high doses of amphetamine. The idea then is that the increased spontaneous transients lead to the increase in striatal dopamine, and the decrease in adaptive transients lead to decreases in reward processing. The increased spontaneous transients are hypothesized to lead to positive symptoms, whereas the decreased adaptive transients through decreased reward processing are hypothesized to lead to negative symptoms. And then if that's the case, the ideal medication for negative symptoms should increase the adaptive transients without at least worsening the spontaneous transients. And then, in fact, that seems to be what the drugs might be doing, but we need more research to show that. For modafinil, does it increase adaptive transients? There's one study that suggests that maybe it does, although there's a little bit of a limitation in terms of the study that I showed you. Does it worsen spontaneous transients? Well, maybe, but we need more research to know for the reasons that I mentioned. Amisulpride at low doses does seem to increase adaptive transients. There's several lines of evidence that are suggestive of that. I couldn't find anything saying whether it does or doesn't worsen spontaneous transients. This is something that would be interesting to test. Okay, so just to conclude then, this is sort of a parsimonious theoretical account with clear predictions with treatment implications. A lot of work still needs to be done to further test the account, and at the same time, hopefully, use these ideas to understand treatments, current treatments, and develop new ones. But the theory, fortunately, provides clear signposts for what needs to be done, which is looking better at this issue of adaptive versus spontaneous transients. In that sense, there's nothing more practical than a good theory. So just to finish then, thanking Michael Frank, with whom I had lots of interesting discussions over the years about this, and who is a co-author on that paper that I mentioned, and very valuable ideas that we've always exchanged, and Vasco, who's a postdoc in my lab, with whom I've discussed many of these issues as well. Thank you very much for your attention.
All right, thank you, Tiago, for that excellent presentation. Our next speaker is Dr. Andreas Meyer-Lindenberg from the Central Institute in Mannheim.
Thank you, Diana. Excited to be virtually here. I'll be talking about computational modeling on the systems level. I'll be applying this to psychotic disorders, and as you will see, be touching upon some of the same issues, for example, relating to dopamine, although on a different level description with a slightly different angle. Now, there are many reasons why you might want to do computational modeling. Here we're approaching this from the point of view of wanting to find new drugs. So we are approaching it from the translational angle towards psychosis. What you can see on this slide is tests of depression, but I guess the same logic applies to psychotic disorders. If, for example, you look at the forced swim test, you see a rodent who has given up swimming in a jar of water and kind of hangs there and looks a little depressed, and from that behavior we would like to infer, and we actually use this to screen for antidepressant drugs, although it is debatable whether a behavior in a different species is really very useful to draw inferences of that kind. And of course that becomes more problematic when you look into human specific disorders such as the psychosis. So there are various ways you can address these preclinical translational problems, but one of them is to try and move away from behavior to the neural systems level, because there are several features, as we will discuss in this talk, that you can look at the systems level that are linked to a psychiatric phenotype and also to the risk of a psychiatric phenotype, and that you can look at translationally. You can look at it in a human brain, but you can also look at it in a rodent brain. And if you're asking about circuits, you can circumvent the question of overt behavior. This slide is showing one phenotype that we've been interested in for quite a while, a connectivity phenotype, like all the phenotypes I'm going to show you today. So we're not looking at a specific region of the brain. We are asking how various regions of the brain connect or interact. This is looking at the connectivity between prefrontal cortex and the hippocampus, a phenotype that a long time ago we found to be linked to psychosis during working memory. We found a few years ago to be linked to genetic risk for psychosis. And if you look at this paper, which is by Josh Gordon in a mouse model, you can see that you can actually measure hippocampal prefrontal connectivity, in this case using electrophysiology, in a genetic high-risk model for psychosis in a mouse. And in this way, you can link between species and you can link between various descriptors by looking connectivity as a phenotype. Now, if you want to make that useful for translation, the various things that you can do, you can make humans behave like rodents. This is an example of this. This is a radial maze of the kind that's being used to test memory processes in rodents. And we have translated that using virtual reality into a corresponding situations for humans. And then we can see, as you can see on the right here, that prefrontal hippocampal connectivity is modulated based on the phase of the task or the memory function that the test taxes in this context. And probably more useful for day-to-day translation, you can do the opposite and try to access the systems level in rodents. This employs rodent or translational neuroimaging. In the examples that I'm going to be showing you, we're using high field MRI. And when you do that, you see that although the rodent brain is a lot smaller than the human brain, you can actually see very similar connected systems in the rodent and in the humans. This is now not looking just at the connection between two brain areas, but looking at well-defined connected systems, like, for example, what's in humans called the resting state network, which you can see here, and an anti-correlated lateral network with this very close correspondence really in the rodent. And the same applies even to the mouse brain, which is still smaller, but you can see with very high precision and a lot of regional homology, similar networks during resting state MRI in a mouse brain. And once you have established that correspondence between species in connectivity phenotypes, you can use this to ask about the underlying computations, topic of our symposium, and you can use this to ask questions about drugs. And I'll be showing you some slides about ketamine, which, as you are aware, by virtue of its NMDA antagonistic effects, is of interest as a model of the disconnection hypothesis of the psychosis, because it induces a reduction of long-range connectivity, while through interneuronal effects boosting short-range connectivity. So things that are similar to what you find in patients with psychosis. And this is just showing you in a very brief outline, with the lines being connections between brain area that you can actually nicely see induced by a ketamine dose, a change in the connectivity between, for example, here, homologs of the human prefrontal cortex and the hippocampal formation in a rodent. And then you can, as a proof of principle, try to do a similar experiments in rodents and in humans to see whether you see similar comparable effects of drugs in both species. This is again looking at prefrontal hippocampal connectivity, comparing a human here and a rodent experiment here. And you can see very, very similar changes, both qualitatively, regionally, and quantitatively, if you give comparable doses of ketamine and compare that to vehicle. Now, it is true that not all features of the brain that are computationally relevant can be immediately captured in those regional connectivity measures. I'd like to show you one that you've also seen in the previous talk, and that's the relationship between prediction error, psychosis, and dopamine. Prediction error is a critical computation that species, humans, rodents do to compare their expectation with them, what they actually observe in the environment. And because a reward prediction error shows that my expectation of my environment, my prediction is different from the outcome that I've actually observed, there's a need for updating it, so that error leads to an updated prediction and it leads to learning. And we know that that is something that is crucially linked to dopamine, especially subcortical dopamine. That's the famous study by Wolfram Schultz, and it's canonical, if you will, computational description as a dopaminergic signal of the deviation of the received outcome and the prediction. So this term here, and there's plenty of evidence showing that this is disorganized in the psychosis. This is an example, a study by Graham Murray, who uses functional MRI to read out a signal corresponding to this one, although this is pure function, and patients and controls seeing either rewarded or neutral pictures. And you can see that corresponding to the reward prediction error theory, normal controls show a strong response to a rewarded signal and no significant response to a neutral stimulus. And in patients, this is profoundly disorganized, in fact, reversed. And this dysregulated prediction errors is one of the, we think, pathophysiological pathways that lead you into psychosis. So to measure this in a rodent is possible, but challenging in humans, not that big of a problem. This is a study looking at subcortical dopamine-related activation in this slide in the ventral striatum in relatives of patients with psychosis. This is known to be dysregulated, is known to be abnormal down in patients with psychosis, and in this study we confirm converging results from several labs that show that this is also true of relatives with psychosis. So this is an intermediate phenotype in our parlance. This is something that is linked to genetic risk and should therefore be explored as being on the pathophysiological cascade. And for that you can turn to a variation of translational neuroimaging again. This is again using ketamine as our legacy drug to induce a psychosis-like connectivity state. And you can see that if you give ketamine compared to placebo, you induce a similar down regulation of the reward response in the ventral striatum in humans. And this is shown again, the same study pretty much up here. And this is comparing this to the rodent, only in this case, we're not using MRI, because in a rodent MRI, usually the animal is sedated and we don't pick up a lot of activity in the striatum. This is using oxygen amperometry in a behaving rat. You can see really relatively comparable ketamine induced, or in this case, sorry, what we're showing here is the task-induced actually change in ventral striatal activity, but we've applied that to ketamine and it gives you similar findings. So you can use connectivity features and regional readouts to get a functional readout that you can apply to a computational understanding of the psychosis. But what I would like to now turn to is a broader based, but by now pretty widely used approach to characterize the entire set of connections, the entire very complex set of connections in the human brain, the entire connectome, to use that parlance, using graph theory. And what you do here is that in compared to just looking at two brain areas or set of brain areas like I've shown you before, you look at every brain area, usually look at every volume element, every voxel, or every pixel, if you're looking at structural images that you can measure, and look at the connectivity. For example, in the example here, just the straight up correlation between every one of these voxels and everyone else, which gives you a square, very big, if you have a lot of voxels, as you usually do, matrix in which you can see connections, brighter dots here indicating a stronger connection between the two brain areas that are shown in the row and the column of that matrix, which is why that matrix, as you can see there, is symmetric along the diagonal. And the idea of graph theory now is to take that very big data set and make it simple by thresholding it and only calling two brain areas connected if their correlation is above that threshold. And in this way, you can turn this matrix into a picture where you just put dots here for all the brain areas that you consider and draw a line if their connection is above that threshold by that method. And that gives you a graph. And it turns out that you can use very powerful mathematical methods to characterize these very simple mathematical objects. That allows you to compare between species and to approach a better understanding of the computational property of the psychosis. And to show you this, to start off, I'm going to show you a translational example. This is again using ketamine in a rodent and a human, but this time we're not asking about the changes in specific regions like the ventral striatum or specific to region connectivities like prefrontal hippocampal. We're looking at the entire graph, and what we're seeing is that both in rats and in humans, we have a reduction in long-range motives, such as this one, and we have an increase in closed motives, such as these two, which are the graph theoretical correlates of the phenomena I've just described to you, which we know from ketamine. It's reduction of long range and boosting of short range connectivity. So you can pick that up using graph theory in this translational setup, and you can compare it in the graphs if you plot homologous brain areas, as you can see here. And you can see, although there's no perfect correspondence, there is actually a relatively nice correspondence between the species in the way these motives are reconfigured under ketamine, so in this model psychotic state, if you will. And that is function. It's a functional graph. It's still descriptive in a way, but you can now push this using new method to put it more into a computational understanding of what the brain is doing while it is exhibiting these graphs. And one approach that's very powerful in here is multi-layer neuroimaging, in which you are looking at these complicated matrices which correspond to these graphs, not just once, but in time windows. And in this way, you can look at the evolution of the graphical motifs on the surface of the brain over time during a neuroimaging experiment. If you will, you can see a little bit like the software running on the surface of the brain, and you can use this to derive a lot of measures from the way these graph theoretical motifs evolves. Like, for example, you can see one motif here going away, but another motif staying comparatively similar. That's computationally very expensive, but informative. And one simple measure you can get there is just asking how flexible is this neural response, how variable, if you will, are these motifs, these graph theoretical motifs, as you look at them over the course of a task in the brain. And when you do that, this is an example from our work in psychosis. You can see that there is a flexibility gradient, a lack of stability, if you will, in schizophrenic patients, shown here in black, being more flexible, less stable than normal controls are. And you can see that healthy human relatives who share the genetic risk with the patients but aren't overtly ill are sort of in the middle. So this is an intermediate phenotype. And in this paper that I'm showing here, we also induced this experimentally, in this case not using ketamine, but using memantine, and found a very similar effect in flexibility through memantine, indicating that this flexibility change might at least be partially induced by an NMDA or a glutamatergic mechanism. So you can move into functional computation, and you can do the same with brain structure. This is a very early paper I did with Danny Bassett and Ed Bullmore, which was one of a set of papers that appeared in 2008 that showed that if you just look at the structural network of the human brain using graph theoretical method, you can actually show it to be disordered in some interesting ways in schizophrenia. Schizophrenic brain shared some of the very basic properties of these networks with the healthy human brain. In particular, both the schizophrenic and the healthy human brain were what's now called small world network, which is a fundamental property of efficient wiring of the human brain, but it was very different in its hierarchical structures. In particular, it had a very different layering and a very different connectivity between neocortical and older and limbic areas of the brain. And to us at the time, that indicated a disorder in the way this neurodevelopmental disorder of psychosis arises. And if that is so, then you know, you are going to reasonably ask two questions: is that functionally relevant? So does that inefficient alternative wiring have functional consequences? Why would we care about that? And how does that inefficient functional wiring arise? And there is an interesting, I think, paper also by or together with Danny Bassett, looking at that from a very unusual, but I think computationally relevant angle by comparing the human brain to a very simple nervous system of C. elegans and looking at it compared to computers. And you could argue that computers are evolved to be very computationally efficient by market forces, the same way that the human brain and also the C. elegans nervous system has been evolved to be very efficient in information processing as required by the species in our evolutionary context. And what Danny showed in this paper is that indeed we found that small worldness, so the way these complex systems were wired in a small world fashion, was actually present in all of these systems. So then she went a step further and looked at a phenomenon called Rentian scaling, which I'm not going to go into the detail here, but basically you can think about as asking the question, how efficiently geometrically is the network packaged onto its container. Now, in the course of, in the case of a computer, that's a 2D wafer on which the chip is being positioned. And in the case of C. elegans or the human brain, this is the skull in which the brain is contained. And it's been known for a while that computers, generation for generation, are actually getting better and better and cramming ever more connections between processing elements onto a wafer, onto a two-dimensional plate, and there is a mathematical limit to how far you can push that, which essentially is basically, you know, you can cram a three-dimensional brain, if you will, into a two-dimensional wafer. That's optimal, and that connection processing element relationship can be measured by a thing called Rentian scaling, which has been known to be present in computers and pushed very near its optimal in the evolution of chips in the context of evolution of chips, if you will. And a similar thing now held in this analysis for the human brain. And you could find that the human brain is optimally positioned within the skull in the same way that a computer network is optimally crammed onto a wafer. Only in this case, we're talking about a three-dimensional wafer, if you will, the context of the skull. And she then went on and showed that something that's been empirically observed for a long time, but had always been kind of a riddle, namely that there is a strong link between gray matter and white matter volume. If you look about a wide range of brains, you can see the opossum here and the sea lion, I guess the elephant here, and humans are somewhere in the middle. That was very linear, but it wasn't clear what the slope of that line in a double logarithmic scaling was explained why. And we now could show that that slope is almost perfectly explained by Rentian scaling, so by the assumption that evolution optimizes this packing of the small world network into a constrained space, which strongly argues, in my view, that this is evolutionarily relevant and therefore is relevant for information processing. How do these changes in the Rentian scale structural network arise in humans? This is something, as a question that you can ask using a method called generative models, in which you basically specify some rules by which networks grow, which you can see in a simplified form. Here you could, for example, specify that neurons that are closer together have a higher likelihood to be wired. You can specify a wiring cost. You can let the network grow, and then you can ask the question whether such a generated network is similar or not with relationship to the human brain. So you can reasonably ask the question of how do these disordered structural networks arise? And graph theory can give us an answer to that also, and that answer comes from an approach which is called a generative model. What you're doing here is you specify the nodes of the network and then you let them grow according to pre-specified rules, connections that are similar to the axons and the synapses then. And then you can play around with various rules. For example, you can make it more or less likely that far away neurons will form a connection. And the network that you grow in this way, you can then compare to what you really observe in schizophrenia. And this is a paper from Ed Bullmore's lab showing that if you do that, you actually find that the observed disordered structural network is actually very well explained by schizophrenic brains having less of a wiring penalty. So it is less of a problem during the neurodevelopment of schizophrenia to find the long-range connections. And that actually explains the observations very well, and therefore gives you an empirical graph theoretical approach to look at the underlying computational framework, if you will. Now, I've shown you a variety of approaches using graph theoretical methods for translation. I would just like to mention at the very end of my talk that one important application of systems level characterization, in addition to finding new drugs and understanding the pathophysiology of the illness, is also in precision medicine, because we know that our current diagnoses are very heterogeneous. Even psychotic disorders reliably diagnosed but quite heterogeneous. And in order to sort these categorical diagnoses into homologous biological subgroups with the goal of then assigning them to specific treatments, we need informative but high dimensional data to parse the pathophysiology. And I've tried to show you in this talk that you can use computational neuroscience with a systems neuroscience twist, applying graph theory to that goal. With that, let me just thank the institutions that have funded this work and my collaborators, especially Urs Braun, Heike Toast, and Natalia Gass and Danny Bassett. Thank you very much.
Thank you, Andreas, for that excellent presentation. We now turn to our last speaker, Dr. Kirsten Ritter from the Charité University in Berlin.
Hello, thank you very much for this nice introduction, and thank you very much for inviting me. It's a great pleasure to present my research here. The title of my talk is From Machine to Deep Learning and Clinical Neuroimaging. And I would like to start with a short motivation. As you know, reading radiological images is a highly sophisticated task requiring advanced education and a lot of experience. And usually those tasks are performed by well-trained medical experts, such as radiologists or neurologists. But a number of studies have shown that those diagnoses made by medical experts strongly depend on individual characteristics of the medical expert, such as age, experience, or perceptual abilities. And the question now is, can computer programs be better in solving some of those tasks? And the hope is that the computer programs are not only more objective, but also more sensitive in identifying subtle disease alterations. And in my talk today, I will talk about different computer programs from machine learning to deep learning. I will give a first short introduction here, and then I will mainly show two applications in Alzheimer's disease and schizophrenia. And I will conclude with the difficulties of translation into clinical routine. So the first important question is, how does a computer learn? So as you might know, there's this broad field of artificial intelligence, but traditionally has mostly been about rule-based learning. So you define a set of rules, give the rules to a computer, and then the computer sticks to the rules in order to come to a certain decision. Machine and deep learning is different. So this is learning without being explicitly programmed. So here the computer learns based on examples. For example, on imaging data of patients with cancer and healthy controls. The main difference between machine learning and deep learning is that we have in machine learning an extra step of feature extraction. The human experts think about features that best describe the input data, and then a relatively simple classification algorithm is used. And deep learning, the representation of the input data is directly learned within the model. And this is actually one of the main reasons why deep learning networks are so successful these days. The second important question here is, how does machine learning work for diagnosing a disease? So usually we have at least two phases, a training phase and a test phase. And the training phase, we have a number of subjects who are characterized by different kinds of data, such as clinical data, neuropsychological data, blood data, and imaging data. And we have a corresponding set of labels, which are here the diagnosis, but can also be symptom severity or treatment outcome. And then a machine learning algorithm is used to learn a mathematical relationship between the data or extracted features on the one hand and the labels on the other hand. And this learned mathematical relationship can then be tested for totally new, unseen test subjects. And by comparing the predicted with the true labels, we can assess the validity of the model and also the significance of the data or features in describing the labels. So this general framework consists of three major components. First, the labels, then the data or extracted features, and third, the model, which is used to learn a mathematical relationship between labels and features. Let's start with the labels. So the labels are most important because they define the research question. And most studies in clinical neuroimaging so far focused on a diagnostic question, some on a prognostic question, and only a very few on predicting treatment response or identifying subtypes or risk groups. The best studied disease is Alzheimer's disease, probably due to kind of clear changes in the brain and also the availability of large open databases. It's quite obvious that the whole framework severely depends on the gold standard of the labels. And this is not only a problem in psychiatric diseases, but also neurological diseases where sometimes guidelines to change or sometimes only post-mortem diagnosis can be made. Second, the features. I've told you about the difference between machine learning and deep learning. So in standard machine learning, a lot of research effort has been put on identifying handcrafted features such as distribution of white and gray matter, volumes of certain brain structures, cortical thickness, or connectivity measures obtained from a functional MRI data. In contrast, in deep learning, no prior feature extraction is necessary because they operate directly on, for example, the structural MRI data. So here, those algorithms try to find their own representation of data and can therefore perform as a real second reader. And the third component are the models, and there are very different models one can use. So there's the difference between classical machine learning and deep learning, and there's a difference between generative and discriminative models. Very popular within the neuroimaging community were support vector machines. Nowadays, more and more are also using deep learning networks. And I just want to show you quickly an example here. So here you see the Bettenhochhaus of Charité University Hospital, where I'm working, and a painting of Van Gogh. And using deep learning, it's possible to transfer the style from the Van Gogh painting to the image of Charité. And this would show you in an intuitive way how those algorithms are capable of extracting relevant image features and use them for further processing. And within this talk, I would like to focus on a specific type of deep learning network, namely convolutional neural networks. So these are deep learning networks optimized for data coming from multiple arrays, such as images or videos. And the main idea here is that little filters are learned and then slided over images in order to extract certain features of the data. And this gives you a hierarchical representation, which is quite interesting. So in the first layers, image intensities are perceived, then edges are detected, then more complex objects are identified, and in the end, for example, whole faces are recognized. And those CNNs have led to state-of-the-art results in many medical imaging applications, including prostate segmentation, lesion segmentation, or identifying cancer in different kinds of images. And those CNNs have also shown to reach or even exceed human-level performance in a number of different imaging tasks, image classification tasks. CNNs have also been used in neuroimaging. So here the input is mostly structured MRI data, and the most studies that exist used data from patients with Alzheimer's disease and healthy controls, so mostly the acne database, but also some studies on other diseases. At the moment, it seems to be that deep learning networks sometimes give a bit better accuracy, but it's not like that you get a huge increase in accuracy. So it's still something under debate. And one of the main problems we have here are small sample sizes. So very successful deep learning networks have been trained on millions of images. And of course, we don't have so many images generally in medical imaging, but in particular not in the neuroimaging domain. So I have at the moment access to UK Biobank data with up to 40,000 images. So this is really, really a lot. But most image, like disease-specific databases have much less data. So usually in the range of a couple of hundreds or thousands. And if you analyze private hospital data, then you usually have less data. There are ways of dealing with small sample sizes. One is data augmentation. So here you enlarge your database by shifting and translating, and transforming your data in different ways. So this is also a form of regularization. So this makes your model more robust. And another very popular method is transfer learning. So here you train your model on some data where you have a lot of data, and then you transfer knowledge from this model to another model, which you train or fine-tune on another data set. And this has been shown to work quite well, even across domains. Another challenge I briefly want to mention here is bias and fairness. So a number of news have reported that machine learning algorithms have been biased mostly towards race or gender. And this is something we have to be very careful about also when analyzing medical images. Of course, it's not about the algorithms that are biased, but humans are biased, and so databases are biased, and those machine learning algorithms are very good in capturing this bias. So, and it's really important to control for this, and also to analyze, for example, classification results with respect to certain subgroups in order to see if the algorithm might be biased. And the third challenge I would like to mention here is the black box criticism, mostly of deep learning networks. So deep learning models usually give relatively good performance, but at the cost of interpretability, because they are very complex and very, very many parameters need to be tuned. And so it's it's not really like learning and concepts, so what humans like, but of course, if you want to use those networks in the medical domain, you need to understand or to like understand why a certain classifier comes to a certain decision in a certain patient. And there are also different methods to deal with this. In image classification, these are, for example, heat map methods. So they indicate the relevance of the input voxel or pixel for the final classification task. And those are also very useful in detecting bias of images. So for example, here the horse has mostly been classified because of the emblem at the bottom. And this is, of course, nothing what we want. But it's really useful to use those methods in order to see if there's any bias in the data. Now I would like to come to the first application, Alzheimer's disease. As you know, this is a neurological disease leading to severe tissue loss in the brain, which is also visible in structural MRI data. Here for the Alzheimer patient, you see enlarged ventricles, cortical atrophy, and hippocampal atrophy. And most machine learning studies and also companies are focusing or have focused on the hippocampal volume, but other machine learning studies also included the whole brain. And here are also already several CNN studies who have led to state-of-the-art performance. But most of those studies did not look into explainability, so how to explain individual network decisions. And we did this in several studies. So the motivation is here that we have a subject with an image, we feed this into a neural network, and then we get as an output 89% or a probability of 89 that this person might have Alzheimer's disease. And just based on this number, it's really difficult to trust it, both from the perspective of the medical expert, but also the patient. But if you give in addition a heat map indicating the relevance of each voxel for the final classification task, then this might be a mean to increase trust. And we developed here a transparent deep learning framework, which consists of 2 steps. So in the first step, we trained the CNN model to discriminate between patients with Alzheimer's disease and healthy controls, and applied it then to test data. And in the second step, we produced heat maps for each subject in the test set. And in this study, for example, we compared different types of computing those heat maps, namely layer-wise relevance propagation and guided propagation. And what you see here is the average of heat maps for the patients with Alzheimer's disease and here for the healthy controls. And as you see, for guided backpropagation, there's almost no difference between both. So this is more a sensitivity analysis indicating what's generally relevant for this network, but it's not indicating what has been important for a particular subject to get classified with AD. But layer-wise relevance propagation, in contrast, does this. So here you see a huge difference between the patients with Alzheimer and healthy controls. And we quantified this a bit further. So here separately for layer-wise relevance propagation and here for guided backpropagation. And what you see here is the density of the relevance scores for different brain regions, and separately for patients with Alzheimer and health controls. And you see that the densities are quite distinct for LRP, but not for guided backpropagation. So here the densities are highly overlapping. And also for LRP, you have much more variability in this relevance scores for the Alzheimer patients. And at the moment, we are trying to use this also to find subclusters in Alzheimer's disease. Now I would like to come to the second topic, mental health and schizophrenia. So on my PhD and postdoc, I mostly focus on neurological disease, in particular Alzheimer's disease, but also multiple sclerosis. And since 1.5 years, I'm now at the Department of Psychiatry and Psychotherapy at Charité Universitätsmedizin, and I'm trying to more move also in this direction. And I just want to give you a short overview here. So, for example, we took part in several machine learning studies. The first was on depression, the PAC 2018 challenge, where the task was to classify between patients with depression and healthy controls. On the test set, we've got an accuracy of 64%. The best team got an accuracy of 65%, so this was rather disappointing. Although the data set was really nice, so it was a really large data set, but we did not have any further clinical information. It might be interesting to further investigate this also with other clinical variables. Then we took part in a challenge on predicting fluid intelligence in children based on structural MRI data. So here we also had access to a very large database, and we also saw how challenging this is to decode fluid intelligence from MRI data. At the moment, we are also analyzing UK Biobank and Connectome data. We also look into connectivity measures here. And the idea is to decode or to find different personality subtypes here. But at the moment, we are first starting to get a good feeling for the data set. And quite new, I'm also part of the new bill Transregio 265 on alcohol use disorder. So we will analyze data sets here. And here I would like to show you some preliminary analysis on private data from Charité. So this was a rather small set, but I want to just show you what we analyzed here. So this data set was provided by Professor Florian Schlagenhauch and Dr. Jakob Kaminski. We had 79 healthy controls and 65 patients with schizophrenia. We analyzed spatially normalized MRI data and segmented gray matter images. This is our machine learning framework. So this looks rather complex because we wanted to have a systematic comparison between classical machine learning methods and deep learning. So as input we had this gray matter probability maps and the skull strip normalized images. We divided our data into training and test set. And based on the training set, we performed cross-validation and selected the best feature extraction and also the best machine learning model. For classical machine learning, we use support vector machines, logistic regression, gradient boosting, and a voting classifier. And for deep learning, a CNN. Those classical machine learning methods were always combined with a feature extraction. And then when we choose the best model, we looked how good are the generalization results. Now, these are the results for the classical machine learning part and only for the validation set, separately for the gray matter data and the normalized images, and separately for different feature extraction methods. What you see here that we've got higher accuracies for using the F-score as a feature extraction and the atlas. And here we compare the different classifiers based on feature extraction using the F-score. And as you see, we've got here a balanced accuracies of about 77%, which is quite good. But when we then try to generalize this to the test set, then we had a huge drop in accuracy. So for the best machine learning analysis, it dropped down to like 61 or 62%. For the deep learning analysis, the drop was not so dramatically, and this was also still significant with an accuracy of 64%. But also rather small. We analyzed this data also with respect to gender and age, because I think it's very different also to see if there's a difference in accuracies between different gender and also different groups. So here we have the results for the gray matter probability maps, and here for the normalized images. And as you see, the accuracies, so the results are not very conclusive on females and males. So sometimes one of the classifiers is better for males and then the other one. For Alzheimer's disease, for instance, we had a more robust finding where we really found that, like with many different machine learning analysis, that we get higher accuracies for women than for men. But what was quite interesting in this data set, that we've got a higher accuracy quite consistently among different classification methods and also among image modalities for a special age group. And this also should show you this kind of the limits of using a relatively small data set, of course. And as you also have said, schizophrenia is a very heterogeneous disease. And of course, this whole heterogeneity cannot be captured by data size sample of 144. And with this, I would like to come to the difficulties of translation into clinical routine. And I think this is mostly about diverging concepts. So machine learning needs lots of data, prefers standardized data, allows well-defined problems, depends on gold standard of labels. As computation expensive, requires methodological expertise, is perceived to be a black box. And in contrast, hospitals protect the data, which is very important, but which also leaves us with small sample sizes, have very heterogeneous data, which makes even more complicated with different MRI scanners, different sequences. And so there a lot of effort has also to put on standardizing the images or to have really large data sets so that the machine learning algorithms can learn over those variability of the different sites. Hospitals usually provide unstructured problems. They have changing diagnostic guidelines as medical knowledge is accumulating. Of course, different data needs to be acquired. They mostly have not yet the right infrastructure for both like data management and processing. They compete with the companies, and of course they require transparency and responsibility. And I think it's very important to try to work on moving these concepts closer together. And I think a real important step is to get and acquire larger data sets. And this might also be very important for schizophrenia. This is not only important for machine learning analysis, but for any kind of statistical analysis. You need more data which also kind of capture the variability in a certain disease. And with this, I would like to end. I would like to thank my team, especially my PhD students and my postdoc, and my master student, Daniel Vargas, who performed the analysis on the schizophrenia data set. And I would like to thank you very much for your attention. And if you have any questions, you're welcome to write me any questions to my email address. Thank you very much.
Thank each of our speakers for really outstanding presentations. And now I'd like to address a few questions, and really anybody on the panel can address these, but I'll sort of start by gearing them towards someone. So Tiago, you presented data suggesting that you could use these computational perspectives to think about ways to target and optimize medications to treat different aspects of schizophrenia. Can you say more about how you would think about that in a more precision medicine context, and the fact that there is a great deal of variability across individuals in their symptom presentation in their course?
Well, it's a fantastic question. So I would say that we're actually lagging behind a lot, even in the basic science of it, to then be able to translate it into the precision medicine. So like I said at one point, it's quite possible that different underlying baseline states in schizophrenia would give rise to different responses to medicine.
Oops, you cut out, Tiago. All right. So let me actually then also put that question in Andreas's hands, because you ended your talk talking about precision medicine. And can you say a little bit more about how you envision that being applied in the domain of psychosis?
Sure. So, I agree with Tiago. It's a good question. You might call it the million-dollar question, but I guess history has told us that there is no such thing as a pink spot, a simple laboratory test, one value that you can meaningfully do to stratify the complexity of the psychosis enough to guide treatment. So that approach will have to be strongly multivariate, and the technologies such as neuroimaging, but also the techniques such as, for example, deep learning, are very powerful in constructing descriptors out of those highly multivariate data set as the recent advances in artificial intelligence, which have largely been through big data trained machine learning algorithms have shown us. Now, what I've been trying to argue in my talk is that one useful input into these kinds of exercises is to use system level features that are informative about the underlying computations. We know that these things are very variable across patients with psychosis. I've shown some data that as a group, they are responsive to genetic risk and that they are reflected in drugs that either induce, in the case of ketamine, or also ameliorate a psychotic state. So they are a priori, I think, useful as inputs into such a precision medicine exercise. And that's the proposal, essentially. We have a normative framework in which we can put in a lot of these data to differentiate subgroups. And so that's what I would do, fully cognizant of the fact that systems level is only one component of an exercise that will also contain genetics and epigenetics and genomics and performance and clinical features.
Great, thank you. Tiago, let's return to you.
Okay, so sorry, my internet is really playing tricks on me today. Yes, the question you had asked, I should probably start from the beginning again, is a great question, and it's difficult to answer because I think we don't have the basic science on it. So we don't know, for example, if you have, let's say you have a hyperdopaminergic animal for some reason. So I think we need to go back to the animal studies to really understand this. Will a psychostimulant like methylphenidate have an effect on improving the transients, the adaptive transients, or will that animal already be in such a state that even a low dose of a psychostimulant would actually be counterproductive to adaptive transients? We just don't know that. And so there's so much that we don't know on the basic science side of things, because I think people haven't been asking these questions. That's where I think the value of a theory comes in, right? Is it guides the questions. People have been asking these questions because they didn't probably seem relevant, or they didn't seem like the most pressing questions. And I would think that we really need to do a lot of this basic science to begin with before we can even think about moving into the precision medicine with patients.
Great, great. And Kirsten, maybe let me change the question a little bit, because I think you're showing really the elegant potential of deep learning and other machine learning methods, but you did note in your talk some of the limitations of the available data sets for being able to address these key questions. What advice would you have going forward for sort of optimal properties or features of new data collection that you think will really help support the use of these machine learning techniques? Should we be designing studies in a different way, capturing patients in a different way, characterizing them in a different way?
Yeah, so this is a very difficult question, and of course every cohort needs to define their own set of rules of how they acquire data and which data is important. And I don't have like a gold way how to do everything, but the important thing is that the people who want to acquire together data, that they agree on some certain MRI sequences, for example, on certain scan parameters, on certain parameters that are important. And one database which is really nice is the UK Biobank data, of course. So they have like good protocols. But there are also other databases like Connectum, which also have very high quality data. So these are already kind of separate data sets. And then it's also the question how to combine this. I think there is no way of like everyone is collecting the same data. So this is not going to work even across hospitals. But it would really help if people would agree on certain MRI sequences and always use the same type of MRI sequences, and then also agree on sharing this data. And I just saw this for the ADNI database. So you can criticize the ADNI databases a lot. Of course, this is also multi-site data and so ever, but it gave a real boost for machine learning analysis in Alzheimer's disease. So it was so helpful. Also, it was not perfect data. So I think any attempt to collect data and make data is really helpful.
Okay, great. So another follow-up question is that, across the different talks, sometimes you all talked about what I would call sort of functional properties of the brain, things that might be able to change on a pretty rapid time scale, whether that be through neurotransmitter release or functional connectivity, but then also sometimes talking about structural properties of the brain, structural connectivity, properties of gray matter and white matter. And I think sometimes it's a little challenging theoretically and empirically how to put those together in terms of, how, if you have a theory about what might be happening at a functional level, how and why does that impact the structural level, and how do you think about ways to best integrate those? So maybe Andreas, I'll throw that question to you first, because you talked both about functional connectivity and structural connectivity in terms of properties of the brain and its organization. How do you think about those relationships in schizophrenia?
That's an excellent question, Diana, and actually had data brought about how to bring it together, but I ran out of time.
Oh, sorry.
It is exactly the right question. And if you continue to use the graph theoretical approach, there is a very elegant technique called graph control theory, in which you can actually explicitly ask, given the structural connectome, what kind of computations happen in functions. So this explicitly ties together the information about the structural connectome with the evolution, the dynamics of the function. And it gives you relatively powerful, incredibly computationally expensive. This stuff really taxes a multiprocessor grid system to a degree that's quite comparable to what nuclear physics or some advanced ray tracing cartoons do. But if you do it, you can actually see that there are normative ways in which you can get from one brain state to the other, given the structural connectivity. And I think that's going to be a very powerful approach in going forward in neuroscience, also in psychosis. There is an early observation that we had together with Danny Bassett in a paper in PNAS, in which we found that the structural connectome of the brain was wired in the way that if you assume that there is an independent oscillator in every node, it would be just what's called the edge of chaos. It would not completely go into a fixed point, which would be like a seizure or something incompatible with mental life. And it wasn't the case that every oscillator was just doing its own thing. It was just at the edge of that. And that was a first indication that there are actually very strong inference that you can make about the computation, about the dynamics in this parlance of the brain by looking at the way it is wired. And using graph or network control theory, we can now tie these strands together explicitly.
I can also say, maybe from a different perspective, not contradictory, but a completely different viewpoint on this question that you asked, which is the neuromodulatory systems are extremely involved in plasticity, right? And so anything that affects neuromodulatory systems like dopamine is going to have huge structural consequences, both in terms of the disorder itself and in terms of the medications and their effect on, for example, the striatum, right? So antipsychotics, for example, are known to increase the volume of the striatum, and they presumably do so through plasticity mechanisms. And so to a large extent, I actually think that the parts of relating the neuromodulatory systems to their consequences in plasticity is going to be one way in which these things play together, which in some cases, they might or not be causally relevant, the structural changes. For example, I remember I was a co-author on a paper when I was a postdoc in which we were looking, it was a cross-sectional study, but nonetheless, we were looking at surface morphology in the basal ganglia of patients with ADHD and the effects of the medication, so the ones that were on medication versus the ones that were not on the medication. And we saw that stimulants would have this big effect on increasing local volumes in places in which they were decreased to begin with. Now, you might be sort of tricked into thinking that this is important, but if you think about the fact that stimulants have such a short period of activation, so if you don't take a stimulant for 48 hours, it's gone from your system and the therapeutic effect is mostly gone, you will immediately think that those plastic changes are probably not really that relevant for the treating of the disorder itself. And it's a clear case in which that point can be made. In other cases, it may be more difficult. But just to say, so in that case, I interpret it as, you know, the patients with ADHD who are on medication can learn all sorts of things that they couldn't before, but that's not what's treating the disorder. And so just to point that sometimes the structural changes might or not be causally relevant for the disorder.
Yeah. Great, thank you. Kirsten, do you have any thoughts about that?
Yeah, so from a machine learning perspective, it's also kind of interesting to combine structural and functional MRI data, and there are also very different ways of how to do this. So you can just concatenate the data, or you can think about more elegant ways of combining those data. We focus at the moment a bit more just on structural MRI data and separately fMRI, because already if you just have one modality, this is kind of complex of analyzing this, and if you then have another additional modality within, then you get so much more complexity on this. So this is why we kind of at the moment more separated always. Also, I'm also always interested in multimodal data integration, and if I have, for example, extracted features from like structural MRI data, functional MRI data, PET data, then it's also really nice to combine this. But on schizophrenia, there's a recent study where they combined structural and functional MRI data also using deep learning, and they found like a slight increase in accuracy when combining both. But functional MRI data was also much better than structural MRI data already, so it was not like a huge benefit of combining those. But it's of course one of the key questions, how to extract the relevant data of each so that you get then a kind of boost in accuracy maybe.
Great, great. Well, I think we will stop here, and I want to again thank each of our wonderful speakers for this excellent presentation and willing to be so flexible with this